Cool. Okay. So this week we have lymph and immune. Yeah, we'll just get into it. Okay, so same announcements. My office hours are 2:30 to 3:30, Academic Achievement Center. Feel free to email me. You can email me like if you're confused on quiz questions too or anything. Yeah, and then access my notes or slides for more resources. Okay, so first part, we're gonna do the lymph system, which is a little bit smaller than the immune system. So here are the parts of the lymphatic system. You have the lymph vessels, which is kind of like their own circulatory system, which contains lymph instead of blood. There are lymphatic capillaries, just like the circulatory system capillaries, and they're integrated closely with blood capillaries. These just kind of pick up filtrate from the blood and then bring it back to the heart. Lymph is this fluid connective tissue that's like plasma drained from the interstitial fluid that comes out of the capillaries. It has ions and proteins, but it lacks anything large that can't fit through the space of a capillary. Here are some characteristics of lymph vessels. They're lined with endothelium, that's simple squamous endothelium, which means that they're like really thin, and they're shaped like oak leaves. They have anchoring filaments between them that are made of collagen fibers. So those are attached to each epithelial cell. And when the vessels are surrounded by fluid, they're allowed to like stretch almost, and they're pulled taut, which kind of, it serves as like, I don't know what they're called, but like they're like strings in between the cells, and then once there's fluid around them, the strings get pulled tight, which allows the cells to kind of like separate and allow things into the lymph vessels, which is how you can think of anchoring filaments. They're blind-ended, which means there's no input. So it's not really like the circulatory system where we have like a constant loop. It's kind of like we're taking things from the capillaries and bringing it back to the heart, and then like it's one way from the capillaries to the heart, and then that's pretty much it. That's where it ends, at the heart. So it's a one-way drainage system. And then there's low pressure in the lymph vessels like veins. So they have valves and use respiratory and anatomical pumps, like we talked about last week, to increase, I didn't finish that sentence, but to help increase pressure and bring lymph back up to the heart against gravity most of the time. So here are some functions of the lymphatic system: to maintain fluid balance by draining extracellular fluid that comes out of the capillaries, and it returns it to the subclavian veins. And then it's closely tied to the immune system because of lymphocytes. So it helps with the production and maturation of lymphocytes, and then lymphocytes are natural killer cells, B cells, and T cells. And then third, the lymphatic system absorbs and transports fats. So it helps transport fats back to the subclavian veins that can't be reabsorbed. So it could enter back into circulation. So I pulled this from the lecture videos that you guys watch. I feel like it's a really good visual of how the lymphatic capillaries are very closely connected with the circulatory system capillaries. It's taking the fluid that's kind of filtered out of the capillaries and bringing it back up to the heart to enter back into circulation. And then along the way, we're filtering out pathogens in like the nodules, so then the immune system can go attack them, type thing. So we're keeping the fluids in balance and also helping the body stay protected by kind of integrating that with the immune system. So here are the two primary organs of the lymphatic system. There's red bone marrow, and that's where hematopoiesis occurs. So that's where the white blood cells are made. And then the thymus is where T cells mature. And T cells can recognize specific antigens, so they're part of the specific immune system we'll talk about later. Yeah. And then the thymus is located like kind of in your chest and between your lungs, so that's a visual there. And then red bone marrow is just in the middle of your like long bones. And also, I just want to add that hematopoiesis forms red blood cells and white blood cells. But for the immune system, we're specifically talking about white blood cells. Yes. And then there are 3 secondary organs. Sorry, that's kind of hard to look at. The first one is the spleen, and this one contains lymphocytes that filter blood pathogens instead of lymph. So this is filtering actual pathogens out of the blood, not from the lymphatic system. And there are 2 parts, red pulp and white pulp. The red pulp destroys damaged and aging red blood cells and platelets. And then the white pulp contains white blood cells that destroy those pathogens. And hematopoiesis occurs in the spleen in the fetus. So that's where blood cells are made in the fetus, in the spleen. Second of the secondary organs for the lymphatic system are the lymph nodes. So these are big clusters of lymphocytes encased by dense connective tissue. So they're kind of at a junction of a bunch of lymphatic vessels coming together, and they filter pathogens from the lymph before returning it to the subclavian vein. So you can think of all the lymph kind of collecting from the capillaries. On the way back up to the subclavian vein, it encounters some lymph nodules, and that's where we're filtering some pathogens out before returning the filter or the lymph back into the blood. So flow through the lymph nodes is very slow due to low pressure, and that's because there's only one efferent vessel or vessel leaving the lymph node, where you have a lot of lymph vessels coming in. So it makes it really slow because you have a bunch of roads coming into the nodule, but only one road leaving, going out. And then this is just kind of a clinical application. Swollen lymph nodes result from pathogens being filtered out. So when your lymph nodes are swelling, it just means that they're working and you have like pathogens, some sort of bacteria or virus that's being filtered in there. So you often have swollen lymph nodes near your nose or mouth because that's where a lot of pathogens enter the body. So they have increased activity, a lot of white blood cell activity like swelling. Yeah. Nodules are similar, but they're smaller and less defined. So they're not encased by connective tissue. And they're in parts of the body that need additional protection. So, for example, the tonsils trap invaders before leaving the GI or respiratory tracts. So they're prone to invasion, so you have a bunch of nodules in the tonsils. And then there are 3 sets in the tonsils, malt, galt, and calt. Yeah. And then that picture at the bottom just kind of shows you the difference. Like, you have a bunch of little nodules that are kind of just like clusters of lymphocytes in tissues that need additional protection, where you can see the difference between the node is kind of in the lymph vessel on the way back to the heart. This is like just in the tissue. Here's a little bit on leukopoiesis. So this is formation of white blood cells, and it always begins in the red bone marrow, in the epiphysis, and in flat bones. So it starts with the hematopoietic stem cell, which is stimulated by colony stimulating factors. So this drives or initiates the formation of white blood cells. And then interleukins are another important signaling molecule that help pick which specific white blood cell is needed. So it helps further specialize the type of white blood cell that's going to be made in leukopoiesis. And then at the bottom of the screen, you can just see like the diversity of white blood cells that the interleukin helps select for based off of the needs of the body. So we just talked about how leukopoiesis always starts in the red bone marrow, but there are two types of white blood cells based off of where they finish being produced. So the first is bone marrow derived white blood cells. These ones finish completely maturing in the red bone marrow. So that's B cells and natural killer cells finish maturing in the red bone marrow. T cells, on the other hand, travel up to the thymus that we talked about. It's like one of the primary organs of the lymphatic system, and they finish maturing there. So only 3 to 5% reach maturity, and then many of them undergo apoptosis due to self-recognition. So the body has a really good regulation of T cell formation. So it's really important that the body has a way of regulating what white blood cells actually get formed, because if white blood cells are recognizing themselves as pathogens, then that's when you have an autoimmune disease, because your body's going to start attacking itself. So that's why it's really important that there's really good regulation of T cell formation in the thymus, because if the T cells were not regulated properly, then the body may initiate an immune response on its own cells. So that's known as negative selection. So we're selecting out the white blood cells that are not properly made or recognizing themselves as pathogenic. And then the mature lymphocytes or white blood cells go to the circulatory system through the lymph nodes and nodules. Okay, so that's everything on the lymph system, pretty much. Any questions or anything you want me to go back over or back to? Yeah.
For the three like the set, is there like a particular order you have to notice?
The three
like the malt, or like is there like a particular order?
No, I don't think so.
No.
No, I think it's just just 3 sets of lymph nodes. Nodules. I would basically just know that those are 3, different types of lymph nodules.
Okay.
That's what I think. And that it's mucosa, gut, and cutaneous. I would know what the M, G, and C stand for too. Okay, so now onto the immune system. which is very closely tied with the lymphatic system we just talked about, because we just talked a lot about white blood cells and attacking pathogens. And the immune system is how we kind of protect ourselves against pathogens. So functions first is to protect from pathogens and also abnormal body cells. So quick definition, pathogens are any microorganism that wants our nutrients and energy and are entering the body. So anything that's in our body that we don't want there. And then abnormal body cells are like cancerous cells that are proliferating at an abnormal rate. The second function is to differentiate self from non-self, which is very similar to the first function, just kind of like identifying abnormal cells that aren't supposed to be there. And then a third function is remembering previous exposures, so it's protected upon second exposure. So it's more efficient the next time. So there are two main divisions of the immune system. First, we have nonspecific or innate, and this is present from birth, so you don't have to learn it. It does not respond to a specific antigen or pathogen, and it has functions in both external and internal defense. So you can think of the nonspecific or innate immune system as kind of being like the first line of defense against like any pathogen. So if there's a bacteria or virus or something, the first line of defense is your skin, because it can't automatically enter into your circulation because your skin's there. And then also mucous membranes are another external defense. These are like in your ears, nose, mouth. It's kind of like sticky substance that tries to like get the pathogen to stick to it so it can't enter your body. And then internal defense for nonspecific or innate immunity include proteolytic enzymes, acids, lysozyme, interferons, and the complement system. So these are all very general systems in your body that will attack any pathogen that's not supposed to be there, regardless of what it is. So for example, your stomach is a very acidic environment. So if any pathogen entered your stomach, the acid is going to help try to like fight off or kill the pathogen. It's not like looking for a specific pathogen like the flu or anything. It will attack any pathogen. Lysozyme is another one to bring up. That's in your saliva. So any pathogen that might enter your mouth, it's going to attack. That's not supposed to be there. Specific or adapted immunity needs to be learned through exposure to produce antibodies for the specific antigen that's on the pathogen. So the response is targeted for specific antigens, and antigens are just kind of like pieces of the pathogen that the body can recognize. Are you guys like good on these terms that I'm using? Okay. So these are your T and B cells that have specific receptors that match up with specific antigens and then can attack the pathogen. And then there are two divisions. Cell-mediated specific immunity involves T cells, and then the humoral response involves B cells and antibodies. Okay, so I talked about this on the other slide a lot already, but the first part of nonspecific immunity is external defense. So these are physical barriers like the skin and mucous membranes. So they're external. They're trapping any microorganisms before they even have a chance to enter the body. Oh, and I mentioned mucous membranes, like trapping the pathogen in the mucous membrane. So it's kind of cool to think about, like, this is why you sneeze, to then expel the pathogen from the mucous membrane. It doesn't just stay there. Or cough. And then internal defenses of the innate immune system. So these are chemical barriers inside the body that are nonspecific, like we talked about before, proteolytic enzymes, acids, lysozyme, interferons. So I talked about acid and lysozyme already, but so I'll mention proteolytic enzymes specifically are in the stomach, and they're nonspecific and help break down pathogens if they get all the way to the stomach. And then interferons are proteins produced by body cells that help protect the body against all viral infections. So any virus that enters the body, interferons are going to attack. And it's not specific because it attacks any virus. And then in green, I have a definition of viruses. Viruses are just packages of DNA or mRNA that come into body cells. And then viruses are the ones that try to like hijack our body cells and try to replicate. So they're kind of just like little packages of DNA, enter our cells and then take over the cell, use all of our cells' resources, and then try to like proliferate in the body. And then also the complement system, which we have some more information on here. So the complement system is an enzymatic cascade of events that enhances or complements antibodies in phagocytic cells of the immune system. So complement proteins are in the plasma and they kind of just help out antibodies in white blood cells, is basically what it is. So there's a lot of words on this slide, but to break it down, there are 3 pathways of the complement system, or 3 ways that complement proteins can be activated. And again, complement proteins just help the antibodies or white blood cells attack pathogens. So there's the classical pathway, alternate pathway, and lectin pathway. So the classical pathway, the way to differentiate that one is that it's activated when it finds an antigen-antibody complex. So that means that there's some sort of pathogen and there's an antibody bound to it already. And then a C1 protein is going to bind to it and then go through a bunch of different changes to become C3, which is then going to activate C3 convertase enzyme. And then C3 is going to break into C3A, C3B, and then C3b will form the membrane attack complex. And the membrane attack complex, it's what's going to poke a big hole in the cell and then lyse the cell, so everything comes out. And then the alternate pathway does not need a pathogen to be initiated. It's initiated by water that does the same thing. It converts C3 into C3A, C3b, and then if the pathogen is there, it will bind C3b and then form the membrane attack complex and kill the pathogen. And then the lectin pathway is activated by a sugar, specifically a mannose on the surface of a pathogen. And then it initiates an enzymatic cascade that again causes C3 to split into C3A and C3b. and then form the membrane attack complex. So basically, they're all the same, but the main difference is what triggers them to start. So the first one is started by the antigen-antibody complex. Alternate pathway is started by water, and the lectin pathway is started by a mannose sugar. And then all of them pretty much do the same thing. Do we have any questions on this before we move on? Okay. And then this is kind of like synthesizing everything we've talked about so far in the non-specific immune system, kind of like a stereotypic response to a pathogen. Is like when you get sick, you get a fever and inflammation. So the fever is caused by pyrogens that are sent from the hypothalamus. And the goal of the body by increasing temperature is to increase the metabolic rate. So we're trying to increase everything going on in the body to try to get the pathogen out is why you get a fever. So for example, like getting the white blood cells to the infection site. It's going to go faster if the body temperature is higher. And then inflammation is caused by histamine, and histamine is released from eosinophils and basophils. So those are just different types of white blood cells. And histamine, so it causes inflammation, which actually is vasodilation, which is widening of the radius of the vessels to try to get as many white blood cells to the injury site as it can. And then chemotaxis is another part of inflammation, which is just chemicals that are sent out at the site to try to attract more white blood cells. And histamine is an example of a chemotatic factor, which basically just means that it's helping attract white blood cells to the site. And then the third part of the stereotypic response is increasing the permeability of capillaries so that more white blood cells can get to the region to help fight the infection. So something to note is that all of these three steps are basically to try to help get white blood cells to the pathogen as fast as possible. And then all the cells involved in this are white blood cells. So here's just a breakdown of the types of white blood cells, the granulocytes and the agranulocytes. Yeah. I don't get too much into these here just because this is a big unit and I can't go into everything, but I know your practicals next week and I would definitely know these for the practical too. I think, if you guys have this unit on the practical. Because I think that comes up. So next we have specific immunity. So we're switching gears from non-specific to recognizing specific pathogens that enter the body. So just to remind you, this one needs to be learned so we can produce antibodies against a specific pathogen. And this involves T and B cells. So here is kind of what's involved. Now we're talking about antigens. So this is a specific part of pathogens that initiates an immune response. So a white blood cell in the body will recognize that there's an antigen and then initiate an immune response. Epitopes are portions of the invading cell that's recognized by the immune system. So it's the specific like amino acids or sugars that are on the pathogens that then triggers the immune response. major histocompatibility complexes, or MHCs. These act as a flag that there's an invader, basically. So it's cells in our body that find a pathogen and then kind of holds up a portion or some sort of signal, so then the body can be like, oh, there's a pathogen here. So there's MHC1, and these ones are recognized by T cells, and MHC2, which are recognized by helper T cells. And that's a very important distinction. I bet that will come up. So know the difference. Know that MHC1 is recognized by T cells, and MHC2 is recognized by helper T cells. And MHC1 is also known as non-professional, or they're displayed by non-professional cells. So they have nuclei, whereas MHC2 are professional. So they don't have nuclei. Those are like your macrophages, dendritic cells. So then there are two divisions of the specific immune system. So we've already talked about non-specific, now we're in specific. And then there are two different types of specific. So cell-mediated uses T cells, and then the humoral response uses B cells and antibodies. So now we're going to talk about the cell-mediated response with T cells. So these recognize an antigen on MHC1, like we just talked about. And then you have the helper T cells. Those recognize antigens on MHC2. And then those go on to activate the cytotoxic T cells that we talked about before. The helper T cells also help to activate the B cells. So helper T cells activate T cells and B cells. Cytotoxic T cells just activate and do their thing. Yes, then you also have the suppressor T cells and memory T cells. The suppressor T cells suppress once we're done with the immune response with inhibitory cytokines. So we're trying to turn the response down once we fought off the pathogen. That's what the suppressor T cells do. And then memory T cells kind of help your body remember this response. Like we said, this is specific immunity or learned. So the body has these memory cells and can fight off the pathogen right away next time that the body's exposed. And now we're into the humoral division. This one is activated by the helper T cells that we just talked about. So it's the second one here. And then these have antibodies on their cell surface. So these antibodies are specific receptors of B cells that recognize the particular antigen on the pathogen. And then they can release the antibodies into the body fluids if they're a mature B cell or a plasma cell. And then once those antibodies are in the fluid of the body, they can neutralize the pathogen by coating it with more antibodies or cause agglutination or precipitation, or it's basically like making a clump so the pathogen can't like do anything. Activates the complement system, So again, those are like those 30 proteins that help the antibodies or white blood cells fight, attract phagocytes, or activate natural killer cells. So again, antibodies are released from the B cells after being activated by the helper T cells for a specific pathogen. And then here is a general overview of everything we've talked about when a pathogen is detected in the body. So then there's also that diagram that I took directly from the lecture. So first response is the neutrophils. So these are the first response to inflammation or damage, and these phagocytize pathogens, which means they just like take them in and destroy them themselves. The pathogen makes it past the neutrophils. There's still more pathogens. So then the natural killer cells survey for more abnormal cells a week later. Macrophages take in some more pathogens about a week after infection was initiated. And then so all of those first three steps are part of non-specific immunity. The fourth step is then the initiation of the specific immune response with the cytotoxic T cells and the helper T cells that recognize the specific pathogen on the major histocompatibility complex, or the MHC that we talked about. And then B cells are activated by the helper T cells. They mature to plasma cells, release antibodies specific to the pathogen, and then the antibodies also help attack the pathogen. And along the way, we get some memory T cells formed. So next time the pathogen comes in, we can more quickly initiate that response and fight the infection. Any questions on anything immune so far? Okay, I'm gonna quiz you guys on something. So cytotoxic T cells, which I've just been referring to as T cells, which major histocompatibility, I don't know if I said the right, complex activates the T cell? Does anyone remember? So there are two, yeah.
MHC1?
Yeah. And then MHC2 activates the helper T cells. And then there are 4 different types of immunity. I think that this will definitely come up, so I would be solid on the difference between these. So how you can think of it is active immunity involves like actively making antibodies to protect your body against an infection. Passive immunity is just receiving antibodies directly into your body. So to make antibodies, you're going to need some sort of exposure that's going to trigger your body to make them. So natural active immunity is you're exposed to a pathogen, you're exposed to someone with a cold, and then your body mounts a response and makes antibodies. So that's natural active immunity. You're making bodies and you're naturally being exposed to the pathogen. Artificial active immunity is when your body mounts a response, produces antibodies, but you were introduced to the pathogen artificially through maybe like a vaccine. So this type of vaccine, you're giving someone a partial part of a pathogen, which is making them make antibodies. So it's artificial active. Natural passive immunity, so now we're in passive means that we're just receiving antibodies directly. Natural means that it's not, like, it's just happening in the it's not really humanly created, I'd say. So this one is when maternal antibodies from the mother are given to the baby during birth or breastfeeding. And then artificial passive uses medical intervention to give someone antibodies to help them fight a specific illness. Does that make sense? Okay, we're almost done. So here's a little representation if that wasn't clear so you can see it better, if that helps. And then I just kind of wanted to connect quickly the lymphatic and immune system so you guys know why we're talking about them together. So I remember when I took this class, I was kind of like, I don't even know why these two things are in the same unit. But the lymphatic system is taking the filtrate kind of from the blood and is then literally filtering it to trap pathogens before giving it back to the blood. So they work very closely together. Here are some other important concepts to review that I just didn't get to put in because we don't have enough time to talk about everything. Yeah. So the activation of both types of specific cells and then antibody structure. And if you need more help on these, like email me or come to office hours and the SI notes are available. Here's a study tip. I put this in that I found from my notes when I took the class. After you study, I kind of like go through and try to like map everything out and how it connects. So you can see I put the two divisions of the immune system, and then from this specific immune system, I went into the cell-mediated and humoral response and kind of put down like the main points of everything to make it kind of make more sense in your brain. Okay, got some questions. Oh, how to log in. Okay, first one is up, which is not a characteristic of lymph vessels. You can choose two. Do you guys see it? Okay, cool. Blind-ended is what we talked about, how the lymph enters the lymph vessels in the capillaries, travels up to the heart, and then gives everything to the heart. Yeah, it's one way. It like the circulatory system is a full circle. It kind of it's blind-ended. It only goes in one direction.
It only lets me click one.
Oh, it does?
Yeah.
Oh, okay. Well, both of those are right that you guys selected. So they don't have high pressure. They have low pressure. That's why they need like valves. And they also use the respiratory and anatomical pumps to help increase pressure. And then they do not have a lot of tight junctions. They have the anchoring filaments that allow them to separate and let fluid in. Oh, sorry. Oh. Where do T cells mature? Nice, yeah. T cells mature in the thymus. Where do B cells mature?
The bone marrow.
Yeah. What are the three secondary organs of the lymphatic system? Okay, I'm going to ask you guys this. What are the two primary organs of the lymphatic system? Does anyone remember? Okay, so the primary organs were red bone marrow and the thymus. Okay, so most of you are getting there. The secondary organs are the nodules, nodes, and spleen. Primary organs, red bone marrow, and thymus. White blood cells that finish maturing in the bone marrow are either this is kind of a trick question. White blood cells, not red. They can't be red. Yes, so B cells or natural killer cells finish in the bone marrow. Red blood cells also do get synthesized in the bone marrow, but this question is specifically asking about white blood cells. T cells and B cells are involved in what type of immunity? I don't know why that X is there, but. And then T cells are a part of what division and B cells are a part of what response? Okay, so these answers are all right. T and B cells are involved in specific or adaptive immunity. Both of those work. Non-specific does not work. Non-specific immunity is again like the skin, mucous membranes, or any like other enzymes in the body that aren't attacking a specific pathogen. T and B cells, remember, recognize the MHC, which is holding up some sort of flag for a specific pathogen. T cells are a part of the cell-mediated cellular cell, all those work, division, where B cells are a part of the humoral response. So humoral means body fluids. So that's because the B cells are releasing antibodies into the body fluids. So intrinsic and extrinsic, I saw, those are a part of non-specific. Those are different types of non-specific immunity. Innate immunity only consists of external barriers of defense. Cool, so that is false. Can anyone give me an example of an internal barrier from innate immunity?
Like the chemical barriers in the stomach.
Yeah. Perfect. Non-specific immunity must be learned due to exposure to pathogens. Yeah, you guys got that one. Specific immunity must be learned due to exposure to pathogens. Does lysozyme work for the innate or adaptive immune response? Why? Nice. So it's a part of the innate immune system because it's non-specific and attacks any bacteria that enters the mouth. As a part of the stereotypic response, what do pyrogens cause? What does histamine cause? Yeah, so pyrogens cause fever, and histamine causes inflammation. So another way to think about these is both of these try to help you get as many white blood cells to the infection site as they can. So increasing fever increases the speed of movement, and then vasodilation helps increase the radius of the vessels to get more white blood cells to the region. Decreasing body temperature would slow things down, and constriction would decrease the amount of white blood cells that got to the region by allowing less things through them. Match the MHC with the cell that responds to its signal. Oh, I forgot I could do that. Yeah, you guys got it. Receiving a vaccine is an example of what type of immunity? Yeah, perfect. Yeah.
What artificial passive immunity do you get? Like, what would be an example?
An example of artificial passive.
Yeah.
Artificial.
The baby.
Active. Yeah. No,
No.
that's natural.
Artificial passive.
Artificial.
Giving someone immunoglobulins.
Yeah. So that would be giving someone antibodies, like in a hospital.
Oh, okay, okay.
Yeah. Mm-hmm. The person sitting next to you has the flu and sneezes. The immune response that your body initiates upon exposure is yes, so natural active. So your body's naturally being exposed to it in the environment and it's causing your body to make antibodies. So it's active. Treating an illness with antibodies is a type of yeah, so that's what we just talked about just now. Artificial passive. Cool. So you guys are free to go. I have this up for my how I studied for this course. If you're looking for a new study strategy. Yeah, have a good week, guys. Rest of your week.